Hello everyone, and thank you for attending today's webinar, Exploratory Biomarker Testing, to qualify or validate the assay. The latest scientific and regulatory considerations for biomarker validation. I'm Sharon Saltz, editor of NeuroCentral at Future Science Group, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Please let us know your thoughts on today's webinar by tweeting us at BioAnalysisZone using the hashtag BZWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, we are delighted to have Dr. Afshin Safavi from BioAgilytix presenting for us today. Dr. Afshin Safavi is a veteran biochemist with extensive experience establishing and leading bioanalytical teams in pharmaceutical, biotechnology, and manufacturing companies, as well as contract research organizations. He is founder of BioAgilytix and is considered an expert in various immunoassay platforms and biomarker analysis. Prior to founding BioAgilytix and acting as the Global Chief Scientific Officer, he was the Director of Ligand Binding and Immunoassay at AI Pharma. At Talecris Biotherapeutics, he led the preclinical and clinical assay development team, building on his experience as a senior scientist at Novex Corporation, GlaxoSmithKline, and iGen International. He obtained his Bachelor of Science in Biochemistry from UCLA, his PhD in Biochemistry from University of Kentucky Medical School, and completed a 2-year postdoctoral assignment at the Emory University Department of Pathology. We invite the audience to submit any questions you may have for our presenter throughout the webinar using the Q&A widget to the right-hand side of your screen. We will pose your questions to Afshin in a live Q&A session after the presentation and then follow up any outstanding questions offline. Thank you again for joining us today, everybody. I'll now hand over to Afshin to start today's presentation.
Thank you, Sharon, and good morning and good afternoon, depending on where you're calling from. I want to start by thanking all of you for calling in and giving me the next probably 45-50 minutes of your time. And for us, we're going to together go through the journey and learn and revisit some of the latest information around the scientific, industry, and regulatory trends that we see in the biomarker field as it pertains to drug development. For today, our general agenda is going to be provide a quick history of the biomarkers, what makes biomarker validation challenging. We will review and compare several of the key biomarker publications. We will be discussing quite a bit of detail the concepts of context of use, fit for purpose, and validation parameters to be tested and evaluated. And of course, as the topic, the main topic for this presentation is how do we define qualification validation? And we will review a few caution and some real-life examples of what we have experienced and share those with you. If I can take maybe about a minute of your time for those of you that are not familiar with BioAgilytix, we're a contract research organization located in Research Triangle Park, North Carolina, as well as in Hamburg, Germany. We've been in business for more than 10 years. We have over 300 employees, and we specialize in large molecule biotherapeutics, peptide, hormones, proteins, monoclonal antibodies, ADCs, and recently the cell therapy, gene therapy, and RNA therapies. We are actually responsible, either fully or partially, for 8 out of the 18 biologics. So 40% of the biologics that were approved in 2018, we work either on part or the entire biological aspects of these projects. We have about 70,000 square foot of lab space in US and about a couple thousand square meter in Europe, and over 1,000 publications, reputable journal. BioAgilytix was founded on biomarker work, and that's the topic for today. We are in a world, and as I work with about 150 plus clients that we have, that everybody, doesn't matter if you're in US, Europe, or in Asia, all the companies are under the pressure of producing more in less time and reduce cost. And one of the areas that we've seen tremendous growth in the past decade is that the companies are relying more and more on using biomarkers to assess efficacy, safety, and the mechanism of action of drugs to get a go or no-go decision early on. Just quickly a little bit about the history of biomarker. I'm not going to go back centuries ago, but I want to start with the 1950s. That's when we see the term biological marker was introduced into the literature. In 1977, we have the biomarker was first used in the literature. In 1980s, we all know that the word biomarker was started being used widespread at meetings, at publication, and it is really in 1998 when the National Institute of Health Biomarker Definition Working Group came up with a biomarker definition, which we all use today, a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathological processes, or pharmacological responses to therapeutic intervention. Today you will hear me, at least for the next several slides, that I will be talking about diagnostic world and the CLSI documents with the diagnostic world and the laboratories that in the hospitals that measure your cholesterol and the general chemistry panel that they use. If you look at the biomarker validation as it was evolving, the evolution sort of started earlier in the clinical lab site. And for a lot of the clinical labs, they started putting again and coming up with the CLSI documents, which is a very comprehensive document and provide guidance for them as what to do and how to operate the clinical laboratories. For us, for a lot of us who were in a biomarker in the PK field, and we eventually went into biomarker assay development, validation, and sample analysis, we actually started, many of us starting using what we knew about the PK validation and PK sample analysis and trying to actually apply that knowledge to biomarker. And that was and is challenging. One of the main differences that we face in the two worlds of the diagnostic versus the R&D assays and the exploratory biomarker that we live in, if you look at the diagnostic world, a lot of the assays are run on these FDA-approved 510K approved kits. These are assays and kits that are fully automated. They're usually on automated analyzers like a Roche Cobas. They have low variability. Since they are automated and they measure a narrow window, the variability is between 1 to 5%. We usually see even, most of the time less than 3%. And they provide high qualified reagent. What I mean by that is that these manufacturers, they are running millions. They generate enough reagents and kit lots to run millions and millions of samples per lot. So what that means is that the antibody is not generated in 200 or 300 microliter of labeling. These are generated in gallons, if not hundreds of gallons of material that is generated at a time. If you look at the R&D assays and the R&D world that most of us live in the drug development, we use these 96 well plate formats. There's high variability. We can see that as much as somewhere between 10 to 25% variability that we see from lot to lot with some of these, and we will go actually, I have an example that I will share with you later on. And the reagents that are generated, they are research-grade reagents. So we take these kits that the manufacturers have put together for research, and we apply our qualification, validation, whatever parameter testing and optimization that we perform in order to be able to use these in support of clinical trials. Another aspect that makes biomarker validation challenging is that in the PK world, we have well-characterized reference material. The C of A that you get it is pretty comprehensive. That is actually the drug that the manufacturer and the sponsor have been using to eventually, dose the patient. So there's quite a bit of information around the purity and all aspects of the reference material that is provided that we can move forward with. The reference materials are usually available, or if not always, is usually available in the pure form, and it is provided in a simple buffer or formulation. In the biomarker world, the recombinant reference material is what is normally used because we don't really have the true material. We cannot go and purify biomarker X from 100 milliliter of blood to go ahead and use that as a reference material. And this recombinant material, we don't truly know if it's a true representation of the endogenous material. It may or may not be. It is usually not available in the pure form, the recombinant or the reference material that we use, and the endogenous form available only in the biological matrices. So this is a concept that I would say one of the most important concepts that I want to go over today. Next time when your clinical team tells you that you got 20 or 30 picogram per ml for a particular biomarker, and why are you getting 10 picogram per ml as a baseline, this might be a helpful example for you. If you know that from a biological perspective you have done everything right and you have gone through the general practices to make sure the assay is running properly and in a controlled fashion, then this is the real explanation for it. The reality is the biomarker kits that we purchase, at best what they provide are relative accuracy. They are relative quantification. And why is that? It's because, again, we're not using a true reference material. These are recombinant protein that a number has been assigned to them. And therefore, one of the key concepts that I hope you get out of today's webinar is that, and one of the things that I hope you can go and actually modify your SOPs if you have not done so, that again, we need to start, when we use these R&D kits and we validate them to support or qualify them, to support biomarker studies for exploratory biomarker phase 1, 2, and beyond. These are really relative accuracy numbers that we are reporting. It doesn't mean it's bad. Still, we're going to be able, if the assay is reproducible and sensitive enough, we can still recognize and be able to report pre versus post-dose changes. Again, but we have to remember those are relative numbers. Let's talk about the second concept that is extremely important, and it's sometimes still overlooked, the concept of parallelism. Parallelism is a demonstration that the sample dilution response curve is parallel to the standard concentration response curve. This is necessary due to the fact that the LBA assays, a binding interaction is being measured rather than intrinsic physiochemical properties of the biomarker. We want to make sure that there's absolutely no apparent bias when we are back calculating the samples after dilution against the curve, and we want to make sure we cover the entire curve range when possible. But the beauty of, again, performing biomarker parallelism for biomarker work is that unlike PK assay, where we have to wait until we actually go through the phase 1 or so to get the samples, the incurred samples to perform parallelism, we can actually perform the parallelism studies early on by purchasing series of normal and disease individual patient samples in a proper matrix. So the true difference here between the dilutional linearity, which many of us are used to, and the parallelism here is that the dilutional linearity, you spike the matrix with the drug and then you serially dilute it. With the parallelism, you actually have to go and find a series of samples with a high endogenous level. And that's sometimes possible and sometimes, unfortunately, it's not possible. And then take those samples and then serially dilute those samples that truly, again, that biomarker represent what should be in the sample eventually, and serially dilute that to establish, again, this dilutional linearity. But again, in this case, you're using endogenous samples to make those dilution and establish the linearity. Another factor that is very important when it comes to biomarker studies biology matters. I don't think I need to go into this into too much detail. We're born different, our adulthood are different, and when we get old, there's a series of transcription factors and biomarkers and proteins that may or may not be there when we started life. And genetically, we're all different. So what else is the problem? The fact is, again, the makeup of a PK scientist and a biomarker scientist are different. Most of the PK scientists, they were analytical scientists that went and started learning, again, the biomarker field. A lot of the biomarker scientists were biologists or biochemists that actually came into the pharmaceuticals or biotechs, and they learned the analytical technique to apply to their biology. So that is why important, and there is not right or wrong, and there's a lot of cross-pollination, and that's the kind of scientist that you need in order to perform the biomarker studies. You got to understand the analytical world, and you got to understand the biology. But most often, what is very helpful is that where we see problems with some companies is that there is maybe sometimes a lack of cross-functional teams. So one of our recommendations is to the clients that when we go start working with them, and we're one of the pieces of the pie, of the puzzle, when we support the biomarker studies, is that make sure you have somebody from every functional group. In particular, the clinical team and the bioanalytical team need to early on open up the conversations. So the biomarker world that we live in, it is pretty confusing. There are all kinds of white papers out there. There are concepts being thrown around. As biological scientists, we like to have a SOP or a guidance that we follow so we make sure we don't miss anything. But that is not the world of biomarker today. And I don't think we will ever end up with an absolute SOP or a guidance that will tell us exactly what to do in the biomarker for biomarker validation. The concepts of fit for purpose and context of use that we're dealing with, the kit-to-kit, the lot-to-lot variability. We're dealing with biomarkers that have, the samples only have high endogenous level. How are we going to generate those samples with a lower level, low QCs? We have samples that there's only low endogenous level. So do we need to perform the parallelism, spike them with recombinant proteins, and doesn't that kill the purpose? What kind of rule do we use in order to come up with acceptance criteria? How about the stability in terms of qualification, quantification, relative quantification? And there are great white papers from WRB, from GCC, from EBF, and various other organizations. So that is why within our teams, within our meetings, and in the company and outside the company, there are good debates and discussion as what to do and what not to do. And today, again, my goal is to review some papers and show you some real-life examples. And maybe sometimes I tell you what not to do, and there are times that I will tell you as what is the industry standard in terms of what we see the most, what people are doing, and what at least scientifically makes sense. There are hundreds of biomarker papers that are published out there. With that being said, I believe there are 4 key biomarkers that if anybody writing their SOP or they are a newcomer in the biomarker field, that I would highly encourage you to read and try to fully understand. And I want to go ahead and actually, I already mentioned BEST to you. That again, that's where you find the FDA NIH biomarker working group that put that together in 2016. I highly recommend for your definitions, use that as your primary reference point. There are 3 key papers, one from FDA and 2 white papers that I want to go ahead and share that with you very quickly. I'm not going to have time to go through every one of these papers in detail. I will touch on them for a few minutes, but I'm hoping that in the future I can actually provide more deeper discussion over the next few months with regard to exactly what is it that every one of these papers are driving. In terms of the comparison of the 3 papers, there is Dr. Jean Lee paper that was published in 2006. A lot of us that started, again, the biomarker field a decade plus ago. this is what we have used as our primary source to start with. In 2018, FDA came with a regulatory guidance paper, and that is something that I will touch on very quickly. And a few months ago, we actually had a paper that I had the pleasure to be one of the authors on this paper. It was a great collaboration between the industry and regulatory agency and the CPAT folks, as well as Duke Margolis. So we have somewhere about 20, 25 people, with about a third of the folks being from CBER and CEDAR that were on this paper. If you look at Dr. Jean Lee's paper, in 2006, there was really not the FDA biological drug assay guidance, nor the CLSI guidance really fully met the need of the drug development biomarker arena. That's what was recognized. And the goal of this working group, which there were about 10 industry leaders, got together, and they came up with a practical approach for biomarker method development validation. And this is when we really see the whole concept of fit for purpose coming to our world, which is a great concept, and we will go into more detail. In this paper, there were discussion around the assay parameters as well as the acceptance criteria. There are a couple key statements that I want to read for you, which I think is the meat of this paper. Acceptance criteria should meet the predefined needs of the study rather than simply reflecting the performance capabilities of the assay. At that time, they did not call it context of use. However, this is really the direction that they were thinking. They also talk about in this paper that we should respect the biological variability of the biomarker within and between the study population. And they talk quite a bit in this paper about the fit for purpose status of the biomarker method. And to me, those are some of the key points that, again, this paper is delivering very nicely. And although the paper really established which parameters to investigate and how to investigate the parameters for biomarker validation, it did not provide a clear guide, a complete clear guide for setting up acceptance criteria. And I'm not here to say that we can ever come up with a clear guide. In 2018, FDA eventually published a guidance paper that was really a discussion that started in 2013. And the information in this guidance really applied to the bioanalytical procedures for GC and for chromatography area, as well as for the ligand binding. And it pertained to the level of drug, to the metabolites, to therapeutic proteins, and biomarkers. There are a couple of things that I'm very happy to see that these were included in the FDA guidance. One is that the concept of fit for purpose. That is very important. That was, again, it started in 2006 in the biomarker world, and it evolved, and you have about 10 plus years later, the fact that FDA puts it in their guidance, this is a very good and big accomplishment for biomarker work. The second concept here is they don't come up and actually tell you exactly what is the acceptance criteria, which parameter to look at, but they do tell you that the PK world is the starting point. What you apply for again, the concept, the parameters that we apply to PK world, that's a starting point. However, I like the fact that they say that's a starting point and it's not a must. And I think one of the key points that they have included in this is the concept of parallelism, which is, a lot of us were very happy to see this in the guidance. So the guidance covered the biomarker validation in about less than a page. There are many things that are missing from this, but at the same time, I view this guidance more as a very, very 10,000-foot general approach, which is, again, in line with what we're doing in the industry anyway. Now, we're going to the 2019 Piccoli et al. Paper, the CPAT paper that I mentioned to you. This was almost a 3-plus-year journey. And the fact that we had, again, a very good representation from folks at CBER and CEDAR, and from 10 to 15 pharmaceuticals and biotechs and a couple of CROs, it allowed us to also go back and look at almost every white paper and guidance paper that was published previously, and take those all into consideration. In addition, we had folks that have used the CLSI guideline. They were involved in writing some of those CLSI guidances. This was very good because this paper really gets into the detail, and I would highly recommend you guys. It's a long paper. The appendix is very long, a lot of examples in the back. I highly recommend you to read it. I do want to make sure that I make one thing clear. The intention of this paper was to come up with a bioanalytical assay validation for clinical biomarker qualification. That is very important. It's for clinical biomarker qualification. The presentation that I'm providing to you is for exploratory biomarkers and what we're talking. So, although this paper does not is not exactly 100% applicable as is to the exploratory phase 1, phase 2 biomarker world that we live in, however, many of the concepts that are put in this paper are very much applicable, and a modified form of these concepts or acceptance criteria, recommendation for acceptance criteria, could be used, should be used as you prepare and modify your SOPs and as you do your qualification or validation. In this paper, it was very clear that and the concept is introduced over and over, that the goal of biomarker assay development and validation is to construct an assay that adequately meets the goal of the investigation. It is quite a bit discussed around that, the fact that biomarkers are endogenous molecules, so they are affected by the physiology, disease biology, pathology, the treatment administered, and the environmental factor. We have to take into account the type of molecules we're measuring and the context in which the biomarker is being applied. If you don't take anything else from this webinar, I think this is a very key important that was stated in this paper that we all got to remember. It is important that the assay procedure and resulting measurements are suitable for the biomarker's intended purpose. The context of use is introduced in paper and discussed in quite a bit of detail. And the biomarker context of use should be proposed early on in the biomarker validation process. Remember earlier on I said the sooner that you actually start, you're in the functional team in order to have a successful biomarker study. And the sooner the clinical team brings the bioanalytical team and the project manager around one table and start talking about and design the context of use together, the chance of success for you and actually generating data that makes sense in the, after completion of all the validation sample analysis, it will tremendously help you. So in this paper, they do mention the most important prerequisite for assay design technology selection is a definition of the context of use of the biomarker. Focus should take into account application requirement, test population, patient population, sample acquisition. And this is this context of use is actually what's going to drive the fit for purpose, and we will talk in a little bit more detail. So, by the way, after this webinar, I know these slides will be, you can register, I believe, on our website or on BioAnalysis website, one of those two, and you can download these slides. And this is a list that I would highly recommend you to have when you do discuss the context of use around the table. The patient-specific characteristic, patient age, patient gender, ethnicity, factors such as exercise, eating, medication, sample storage, sample time, collection tube, and so on. You have this list. Now, I know I have a lot of people that are calling in and have called into a webinar from the contract research organization, and sometimes we're not privy to all the conversation and discussion that are going on. You might get a client coming to you and say, here's this assay, just buy this kit and validate it. But I would highly recommend, and this is something that we highly recommend to our own internal scientists, and we try to encourage it, try to make sure the client understands that this is really a partnership when it comes to developing these biomarker validation and sample analysis. It's just not a black box. And the more information that we know, that we discuss, and they're willing to share this information with us, we make sure that the validation is done properly to meet these needs. So that's something that, again, if you're an external bioanalytical lab or internal bioanalytical lab, make sure you have these discussions. Now let's talk about the context of use and fit for purpose. So once you have the context of use, and you know through that what is the required sensitivity, the type of samples you're going to get, you may even have some information during that time around the pre versus post dose changes you may see. Is the level of drug going to be high, and do we need to look at the drug interaction potentially with the biomarker that you're measuring? A lot of factors that you can learn during the COU. At that point, you come up with your fit for purpose validation. And this fit for purpose validation, you pick parameters that is going to qualify or validate. You're going to look into those parameters in order to actually eventually generate data that's going back and be able to support and provide answers for you to make decisions. Now, let's talk about, again, some of these parameters quickly. The panels that you see on the left side, these are the ones that we are familiar from the PK world, or just a standard validation for criteria that we look at. The standard precision accuracy, QC precision accuracy, calibration rate, range intra and intra-assay precision accuracy, parallelism, specificity, selectivity, stability, and so on. The reason I scratched dilutional linearity, because I want to make a point here, and I want to go back to the concept of parallelism. Please note that, again, aside from reproducibility, sensitivity, or the lower limit of quantification, parallelism is one of the top 3, at least in my book, parameters that you have to establish and evaluate to actually have a decent assay, and it's you know enough about it to move it forward. So this concept, again, you need to really pay special attention to parallelism. Parallelism is very much tied to the minimum required dilution that you have to establish early on. In the event that you cannot establish parallelism, you cannot find high endogenous level, there are instances that you have no other choice than to perform just dilutional linearity. With that being said, as soon as you go through your trial and there are endogenous samples available, you need to amend your qualification validation and again, perform parallelism studies. So let's talk about a little bit about, again, the panel on the right side. We already talked about the concept of relative accuracy. I think I've overstressed that reproducibility is one of the key factors. The LOD, LOQ, those are obviously ULOQ, LLOQ, those are parameters that we always measure. We already talked about parallelism, specificity, selectivity, stability. Those are important factors that need to be measured. But depending, again, your context of use, do you need to really measure absolute accuracy? If you have to measure absolute accuracy, that is a very, very difficult process for the biomarker world. And this is a concept that sometimes even the QAs, the quality assurance unit within some of the big organizations or small organizations, they are not fully aware that, again, we're using recombinant protein, not sometimes, again, the absolute, and we cannot really come up with absolute accuracy. Robustness, ruggedness, and drug interference. So let's talk about quickly around the qualification versus validation. The reason I mention everything that I mentioned so far, because I want to bring this concept of qualification versus validation home for the biomarker. The reality is there's confusion. And there are companies that they check parameters and they call it assay validation, and somebody else might call that a qualified assay, and vice versa. The confusion really stems from the diverse scientific background that we all come from. If you have scientists that are coming from manufacturing background, they have a clear idea. They know exactly, and you can go from company to company And 95% of the time, the parameter, the procedure they use for what they call assay qualification or qualified assay for manufacturing support versus a GMP validated assay for product release are very different. There is a clear distinction from a regulatory perspective and from industry and from all of us that we work in that arena. But when it comes to biomarker arena, there is no clear line. And so I can tell you what we have done in our company and that has to clear this. And this is something we have shared with many of our clients, and they're using this. The table that I show you, it works for us and for many of our clients. For your company, depending on company culture, the level of risk you're willing to take, or how early you want to go proceed with qualification versus validation, this may vary, but I encourage you to establish something like this within your company. For exploratory biomarker, again, we use assay qualification. We look at truly the precision, accuracy, upper and lower limit of quantification, and we perform parallelism. Those are very important factors that we include in our exploratory biomarker. We run those usually over 3 runs. For our validation, it is almost like a PK validation with couple big differences. One, we look at a series of endogenous samples to find low, mid, high quality control that again of endogenous nature, so we can use those. Parallelism, again, by the time we get to validation, has already been looked at multiple times, many times, but we try to find as many samples and include during the parallelism studies. And we actually, again, remember, we talked about the relative accuracy. We want to make sure what becomes important is the reproducibility. The assay reproducibility becomes very important, and we try to run minimum of 6 runs to establish many of those parameters. For unit qualification, when we look at, again, these short-term, long-term stability, freestyle stability, those are recommended. But again, we look at the context of use to see and what we know about the biomarker. Stability, if you're just going to go and you do the assay qualification, it is client dependent. We usually recommend it, but again, it's client dependent. When it comes to what in our company, what we call assay validation, every one of those parameters will be looked at in detail. Now, I want to go ahead over the next 10, 15 minutes that we have. I want to share with you some real life examples. And this is not to scare you, but to bring you some awareness. And if you're dealing with some of these issues, I think then you don't feel at least lonely or alone. Let's talk about multiplexing. It's a beautiful word. We all use it. We've been using it probably since 1999, since 2000, when we all got the direction to squeeze more from every sample. There is a balancing act. We have to make sure that we balance the sample volume. Yep, we want to use a small sample volume. Time, yep, we want to go ahead and proceed and do as many analyses in a short time. And yeah, we want to keep the cost down. But if that is come at the expense of quality, it is going to come back to hurt you. So for us, one of the key factors that we always stress and focus, the context of use, the fit for purpose, and we got to make sure, depending on those two concepts, what level of quality needed in support to support this study. I already talked to you about parallelism, and I don't expect you to be able to read this slide. But let me give you the summary. It is nice to be able to use a large plex and generate a lot of data. But this is an example of a client came to us that they have generated millions of pieces of data on this for exploratory work on this panel, and they came to us to validate. In general, we don't really validate anything that is over 10 plexes. Just the biology of the interaction and crosstalk between these antibodies makes it very different. So from a technology and platform, it might be very nice, and it's doable to go to very large plexes. But from a biological perspective and the molecular interaction that we deal with, that is something that can become problematic. It doesn't mean the large plexes are bad. It just we have to be careful with them. For this particular project, after basically running series of parallelism studies, we found 25 out of 45 biomarkers did not show any parallelism. 7 biomarkers require 1 to 4 dilution. 11 require 1 to 8. 2 require 1 to 32. And by the way, if you dilute the samples to those MRDs, all the results that you get from clinical studies is going to be 0, below the limit of quantification. What this means, if you were actually using this 45-plex, the data, the samples that you got numbers for are, for lack of a better word, are garbage. They don't mean anything. And the samples that you got 0, they're the ones that actually are the ones that, unfortunately they were diluted under the sensitivity level of the assay. So the short of it, you can be spending hundreds of thousands of dollars running the assay. At the end of the day, it doesn't give you anything. And that is why there are a lot of experts out there that they believe over 90% of the biomarker data that has been put into the literature for a variety of reasons, not just this reason, in the literature is non-reproducible. I want to talk to you very quickly about the lot-to-lot variability. This is an example of a project that we ran. The client came to us. They wanted to measure 11 biomarkers. We split based on our knowledge and talking to the vendor. We put them into a 8-plex as well as 3 single-plex. The client thought that this project will be, they will be able to recruit all the patients within 1 year. It turned out 3 years to recruit the patients. It's a very, this client is very, I would say, quality driven. And we actually talked to them and we just discussed that let's go ahead and put buy additional kits for every lot that we buy, so we can go ahead and run series of low, mid, high quality endogenous quality controls on the kits that we bought and we're running out versus a new lot that is coming on board. And remember, most of these lots, the kit lots, are good for about 9 to 12 months, sometimes 16 months. So you have to plan ahead if your clinical trial goes for multi-year. The short of it is, we saw in some instances these biomarkers were holding ground pretty nicely, no change within 3 years in 4 different lots. But in some cases, you can see we almost see a 200% change, twofold change in just the variability from these kits. What does that mean? Let me just maybe show you an example here. What that means here is that if we actually did not put this bridging mechanism and a correction factor, and for doing that, you've got to work with your statistician within the company closely. If you look at the panel on the left, the MCP2, if the client was looking for decreasing signal, they would have been jumping up and down. They say, look at this, my drug is working. I went from 350 picogram per ml to 100 picogram per ml. And if we were looking for increasing signal due to your treatment, you will be scratching your head saying that I don't understand, or you will be blaming your biological lab or the CRO saying that, wait a minute, what's going on here? I'm here to tell you that this has nothing to do with your drug. This has to do purely with lot-to-lot variability. And if you do not keep close eyes under the lot-to-lot variability and have some kind of a mechanism in terms of being able to bridge those lots together, you will end up with data that can be very confusing. So where we are today, right now, what is happening right now? There was a period that the drug development PK world was going on their own. You had the clinical diagnostic, CLIA, CLSI folks were going on their own. And I would say in the past few years, we have started respecting each other, saying that actually, these guidances that are, although developed in 2 different worlds, they can help each other quite a bit. And I think that's something that the CPAP paper has done very nicely in 2019. And again, I hope that sometime in the future, I can go ahead and have a separate webinar and go into the detail of that CPAP paper with you guys. But we have actually, this paper has managed to merge some of this information together. And there have also been quite a bit of cross-pollination between some of the folks from the diagnostic world now coming into the biomarker world, the traditional PK world. And because of that cross-pollination, we see some of the parameters that are being looked at, the way they're being looked at, the acceptance criteria, the number of the replicates that are done, and the whole concept of fit for purpose is well accepted concept now for the past decade plus. And by addition of this context of use, which I hope I see the industry over the next couple of years adopts it more and more. We now have merged this, a lot of the confusion that I mentioned to you, and I feel really good about where we are. Being in this field for over 20 years, I would say the improvement that I've seen where we are today in the past few years is very promising. So just a couple final thoughts that I want to bring up here. And this is truly from Dr. Jean Lee's paper. And again, more stress was put on this in CPAT Piccoli's paper. At the end of it, whatever you want to call it, qualification or validation, whatever parameters that you decide based on fit for purpose and context of use to evaluate, the number of the replicate you decide to include in your study, you want to make sure that the assay you come up with, it is capable of discriminating changes that are statistically significant. And I want to give you just a quick example. If you are looking for a pre versus post change of, let's say, 300%, does it really matter if your total error is 20%, 40%, or 60%, or 80%? The reality is, at that 300% pre versus post dose, if you're running enough samples, you will see the changes. Now, of course, you don't want to come up with some crazy total error, but at the end of it, if that's what the assay is capable of doing, and that's the only assay you have, it's going to work for you. On the same token, if you have an assay that you're patting yourself in the back and you have only 20% total error, and the pre versus post dose change in your biomarker is only 5%, good luck. You will never ever be able to actually get meaningful data. So keep that in mind, and I hope you can see how I'm tying this back up to the context of use. The second parameter is that biology matters. So when you come up with your acceptance criteria, today we didn't have enough time to talk about acceptance criteria. For future webinars, I hope that I can include that and go into quite a bit of detail. But for today, if you look at it, biomarkers with a high degree of biological variability, right? And if you're dealing with low amplitude of response due to stimulus, what I said, you have only 10, 20% pre versus post dose. These assays require assays that are actually have a very, very relatively low total error, and vice versa. If you don't do anything else, I would highly recommend take this slide and the previous slide and make sure everybody within your organization fully understands these two concepts. And the last thought here, that there is no clear textbook. At the end of it, you have to go ahead and put your science hat on, use your brain when you're dealing with these biomarker validation, keeping in mind biology matters. And one of the reasons that, again, a lot of the biological scientists, they go into the biomarker field, it's actually that freedom that you can go ahead and you combine that biology, the biology knowledge, with the biological aspects to move forward. So with that being said, I believe we have about 10 minutes left, and I want to, again, thank everybody on the phone. I think we had somewhere in excess of 400 people that registered for the webinar from 3 continents, so thank you very much. I want to go ahead and open it up for any questions that you have submitted now. Thank you.
Thank you for that very insightful presentation, Afshin. I hope our audience found it of great interest and value. So yeah, if you do have any questions for Afshin, please continue to submit them using the Q&A tool on the right-hand side of your screen. So at the moment, we've received a number of questions, and I'll start to address these now live on air. So question number 1. If you are asked to find and evaluate few kits to decide which one will best support your study, what is the most efficient way to proceed? Where do you start?
Great question, and I think that's one that is probably in, we're dealing with that. Doesn't matter if you're a CRO in a, or in a biological, pharma, biotech. This is what we deal with. For us, we like to we recommend start with meeting with the clinical lead, with the clinical team. to learn as much as you can about the context of the use for the assay. And we discussed that quite a bit during the webinar. See what level of sensitivity is needed, learn about the patient population being studied, the duration of the study, the sample analysis storage. Again, going back to the slide that I had with about 12 or 13 elements. Learn as much as you can about that. The next step is go and do a literature search and try. You don't want to reinvent the wheel. So do a literature search, try to see what kind of kits and reagents and antibodies and reference material people have used for a similar, for that biomarker. It might have been in a different population, or it might have been even for a different indication, but that is okay. That's a good starting point. Once you identify your 2, 3, 4 kits, we recommend order somewhere about 20 normal samples. Of course, if you can get your hand on the disease samples, that would be great, but 99% of the time, we don't have that luxury. So we get some normal samples, and we start our key experiments or kit evaluation by running parallelism studies. And by running those series of parallelism studies, you will start getting some sense in terms of the assay sensitivity, the assay performance, looking at, again, what is the CV. If you were using some of those endogenous samples as a quality control, what would be the CV associated with those? And that should provide you enough information through really probably a week of work on this, should provide you about enough information on those kits so you can say which one of those you want to go in and further evaluate and take into either qualification or validation, whatever that you want to take it to. Thank you.
Okay, that's great. Question number 2, does the number of samples that need to be analyzed impact the level of validation? Let's say one clinical trial requires 30 samples to be analyzed and another 3,000 samples.
Okay, great question. Look, the short answer is no. It is what you want to do with the data generated, and I want to go back, and the context of use that really will dictate the level of validation you want to do. It's not the sample number. There is a but here. From an economic point of view, you may say, I spent so much money generating the 3,000 samples than the study that generated only 30 samples that I want to minimize any surprises during the sample analysis. And therefore, we look at more parameters, and each parameters will be looked at more extensively. So that is really coming from more from a risk management perspective, and the level of risk that a company is willing to take, as well as, I would say, your budget. So the more you do, the better it is, but at the same time, it's a balancing act. So again, the answer to your question, is no, but I hope the but also provided some clarity around the decision making.
Okay, that's great. And question number 3 is, just like the slide you presented, we have noticed parallelism issues with many of the larger multiplex kits. What is your approach when you get to this situation?
Okay, I anticipate this viewer have seen some of the things that I showed, so I appreciate the question. The lack of parallelism could be due to a couple of factors. There may be something about the biology of the biomarker, like interaction with other proteins at certain concentration or condition, that may interfere with the capture and or detection antibody. The lack of parallelism could also be due to poor optimization of the assay during the development. The reality is, when it comes to biomarker and these multiplexes, when you get more than 10 or so biomarker, the kit optimization is viewed and performed such that every analyte in the kit can be measured, but the assay condition used for the entire kit optimization may not be the best condition for each individual biomarker. So again, this is one of those situations that there is not one clear approach. But what I would, if my scientists come to me internally with this, what I recommend to them are the following, maybe a couple approaches. I may suggest, to just split the biomarkers that are problematic into its own series of a small multiplex or singleplex format, and then optimize it under appropriate MRD to remove and minimize the parallelism issue. Now, let's say you do split these biomarkers from the panel and you still have parallelism issues. Then you could play with conditions such as acid-based treatment to see if the lack of parallelism is due to some unknown protein, lipid, or other type of interaction. So I've seen examples of, for example, this really amazed me that in one instance, a very harsh acid treatment of a sample prior to analysis resulted in regaining parallelism, and we actually ended up improving the lower limit of quantification by almost 10-fold. So if you see parallelism issue, sometimes it is not a bad idea to play a bit with some of the sample treatment condition. And by the way, that is one of the reasons that parallelism should be looked at, and I kept saying that it should have been looked at early on during your assay development or kit evaluation. Try to include that early on.
Okay, thank you, Afshin. So our fourth question is, do you have any openings in your biomarker team?
Well, I can tell you this is my favorite question, I guess, of the ones that have been asked so far. So I appreciate that question. Look, the answer is we're always looking for. First of all, whoever asked that question, please send me a personal email, and I would love to look at your resume. And we're always looking for smart, enthusiastic scientists to hire, as long as you're willing to work hard and you're a team player and you want to, again, as I said, use your brain to do biomarker work. Absolutely, I'd love to get your resume. So yes, please send it. You can actually go to our website. It's bioagilytix.com, and again, send your resume to our HR or directly send it to me. I appreciate that question.
That's great, Afshin. Thank you very much. I'm afraid that's all we have time for today, so we'll address all of the other questions offline. So if you do have any other questions for Afshin, please do feel free to send them in the following email address. So that's webinars@bioanalysis-zone.com. So I'd like to once again thank our wonderful presenter, Afshin, as well as you, our audience and listeners, for your time and questions. If you would like to let us know your thoughts on today's webinar, then feel free to complete our feedback survey in the resource widget or tweet us at BioAnalysisZone. Don't forget to visit us at www.bioanalysis-zone.com for more webinars. Thank you again for attending, and goodbye.